In this lecture, we are going to be discussing the neurotransmitters of the autonomic nervous system. We start off writing sympathetic neurotransmitters on the left, parasympathetic neurotransmitters on the right. And we'll start off on the parasympathetic side because, as was true in the last video, it's just a much simpler branch of the autonomic nervous system to deal with is the parasympathetic side because there aren't exceptions. Everything pretty much works the same way. So we're going to draw a rudimentary brainstem here with the midbrain labeled up above and the medulla labeled down below, the medulla oblongata. We're going to skip down and we're going to draw kind of a half cross section of a spinal cord. So far, this is looking a lot like the previous video where we're going to be drawing a small dot inside of the brain there, one for the midbrain, and this time just two for the medulla, with a long preganglionic neuron and a short postganglionic neuron, both of them releasing acetylcholine. That makes both of these neurons cholinergic. And again, we're going to draw two from the medulla. We're going to draw one long preganglionic neuron, one short postganglionic neuron. Just so you know, the actual nerve that's represented in the nerve above is going to be the cranial nerve 3 or the ocular motor nerve. There's actually two nerves represented in that second neuron there. That would be the facial nerve, cranial nerve 7, and the glossopharyngeal nerve, cranial nerve 9. I'm going to put a dot down below that's going to be the vagus nerve neuron representation, but we'll take a look at that in a moment. Okay, skipping down to the spinal cord then, it is S2, S4, the sacral region of the spinal cord. I always draw the little dot in the center as the central canal, and then I'm representing the gray matter surrounding it with one little jut sticking off to the side, which is the lateral gray horn, a small amount of gray matter that is the exclusive purview, I think. Only the structures of the autonomic nervous system actually use that lateral gray horn. And again, a long preganglionic neuron, a short postganglionic neuron. Both of them are going to be cholinergic. Both of them release acetylcholine. Now, the three neurons representing here are going to be innervating various targets. For example, the very top set of neurons that are shown here would be innervating the eyes. The bottom set that is shown here, the ones that go from the sacral nerve, they would be that go through the sacral nerves, they would actually be innervating things like the urinary bladder and the genitals and some of the abdominal organs as well. But now we're going to be, so I said targets generally for those, but I actually want to talk about a specific target that is involved with cranial nerve 10, the vagus nerve. So I'll kind of draw some of the branches. The vagus nerve is a biggie. Remember, about 100,000 neurons inside of there. So when we draw one neuron, it's just such a misrepresentation of what actually happens there. It's a massive amount of innervation going on. So I'll draw a few branches and a few endpoints here just to show you. They'll be going to the abdominal organs and to the lungs. And this specific one that we're drawing here, and we're labeling again cholinergic, acetylcholine, both for the long preganglionic neuron and for the short postganglionic neuron, they're both cholinergic. But we're actually going to draw the target in this case. We're going to draw one example of the target, and there it is. There's a cell that this particular neuron is actually innervating. Just to draw our key here, a cholinergic neuron we're drawing in blue. And what makes a cholinergic neuron is that it releases acetylcholine. So, since these are cholinergic neurons, the receptors that receive the information are cholinergic receptors. That is what the receptors are called as well. The neuron is called cholinergic because it produces acetylcholine. The receptors are called cholinergic because they receive acetylcholine, and we're going to be using blue for all this stuff today, make it simple. So for example, this preganglionic neuron, talking to this postganglionic neuron that we're showing here, this synapse is going to be a cholinergic synapse. The presynaptic cell releases acetylcholine. The postsynaptic cell receives the acetylcholine. Therefore, the presynaptic cell is called cholinergic cell, and the postganglionic cell will have cholinergic receptors. And the same here for this target structure. Remember, this is not a neuron receiving this information, but it will have receptors for the information, for that neurotransmitter. So a cholinergic neuron, in this case, releasing acetylcholine, yes, it's contacting a target, which we'll talk about what that target is in a moment, but it's going to have cholinergic receptors that receive that acetylcholine. Otherwise, it would never be able to receive input from these neurons, would it? So let's talk about the cholinergic receptors. There are two basic types of cholinergic receptors, nicotinic and muscarinic. The nicotinic receptors are very simple. They behave just as neurons that we've studied in the past behave. In this case, when acetylcholine binds to nicotinic cholinergic receptors, it's always excitatory because it is always a really simple interaction where sodium ions influx. And since sodium ions are positively charged, influxing into the cell, which usually is deficient in sodium ions, so they're passing from an area where they're highly concentrated into an area where they are less concentrated, following their diffusion gradient. This is always an excitatory interaction. So nicotinic cholinergic receptors are always excitatory and they always work the same way. And these, I know there's a lot of information coming your way, but the nicotinic cholinergic receptors are the ones that are found in the synapse between the preganglionic and the postganglionic neuron. So these are excitatory, they're fast, and basically if a preganglionic neuron experiences a signal, it's quickly passed on in an excitatory manner and in a simple manner to the postganglionic cell. The story is a bit more complex with the muscarinic cholinergic receptors. Muscarinic receptors are going to be working through a second messenger system or a G protein coupled receptor system. And therefore, what they do is they make some kind of changes within the cell. They tend to be a little slower to work, but they also are quite powerful. And so these could be excitatory or inhibitory depending on what they do inside of the cell, depending on the cellular dynamics that they start to change. So for example, the M1 muscarinic cholinergic receptor, the M1 tends to be excitatory, and the M2 cholinergic receptors tend to be inhibitory. And the examples that we give here, there's an M1 receptor that is found on the salivary glands. So if it's an M1 receptor coming from the parasympathetic side, you may recall that the salivary glands tend to be excited by the parasympathetic side, and they increase their secretion of fluid saliva in response to parasympathetic innervation. Whereas what we're actually looking at here, the interaction of the target cell up above there, is the interaction on the SA node, which actually is inhibitory coming from the parasympathetic side. So let's take a look at that. The cell here, it's going to be called an autorhythmic cell. The autorhythmic cell is found inside of the heart. These are the natural pacemakers of the heart. And the specific one that we're looking at is found in the SA node, although there are cholinergic receptors on various parts of the heart, especially the electrical system inside of the heart. The SA node is definitely the most impactful of the innervations because the SA node is that natural pacemaker of the heart. So the heart tends to beat at 100 beats per minute, which that's kind of its natural rhythm. But the parasympathetic nervous system actually slows it down to about, say, between 60 and 80 at rest. So the parasympathetic nervous system is in charge and tends to slow down using acetylcholine at the muscarinic M2 cholinergic receptors. All right, let's get to the exciting side of things. We look at our sympathetic nervous system. Again, we're going to be drawing half of a spinal cord. In this case, it's T1 through L2, all of the thoracic nerves, and then both of the lumbar nerve, and then the top two lumbar nerves, L1 and L2. And then this brings us back to kind of what are the nicknames of each of these branches of the ANS. And it turns out the parasympathetic side, if you think about it, remember that we're using a portion of the cranium, a portion of the brain in the midbrain and the medulla, and we're using a portion of the sacral nerves of the spinal cord, and therefore this is called the craniosacral branch of the ANS. The sympathetic side is called the thoracolumbar branch of the ANS because it uses all of the thoracic nerves and then two of the lumbar nerves. Well, let's take a look then. Again, we're using our lateral gray horn here. Some of the gray matter just kind of jutting off to the lateral lateral edge there. And we're going to go ahead and use our blue marker, and we're going to draw two short preganglionic neurons, and then that one special long preganglionic neuron. And let's go ahead and investigate the long one first. So we're going to be going right down into the adrenal gland. The adrenal gland sits on top of the kidneys. Adrenal on top of the kidneys. We're specifically looking at the adrenal medulla. The outer part is called the cortex. The inside is the middle or the medulla. And so we have these blue or cholinergic neurons that release acetylcholine. But how does this work? So we've used these cholinergic neurons, but we're producing a red substance, which in the last video we used to convey the impression that some kind of either norepinephrine or epinephrine is being produced. And that can look really confusing. So let's talk about that for a moment. Yes, these are cholinergic neurons that are innervating the adrenal medulla. But it turns out the goal of the adrenal medulla, pretty much the only thing that it does, is produces epinephrine and norepinephrine, also known as adrenaline and noradrenaline. And so let's take a closer look at how that works. So as we did on the parasympathetic side, we're going to be taking a closer look at the actual synapse itself. And so what we have is a cholinergic neuron, preganglionic, presynaptic, that produces and releases acetylcholine. And then we have these specialized cells called chromaffin cells inside of the adrenal medulla. And the chromaffin cells, their job is actually to produce epinephrine and norepinephrine, but they have cholinergic receptors. So it's possible that you can use a cholinergic input to enhance the effect of an adrenergic cell, if you will. So the end result is that, the adrenal medulla, this particular connection is extremely powerful because as the chromaffin cells are induced to create epinephrine, about 80 to 90% epinephrine, about 10 to 20% norepinephrine. They're close cousins. They both have very similar effects. As these cells produce that stuff, it actually gets put directly into the bloodstream, and we're showing a vein here, and we're showing the stuff entering into the bloodstream and going off to cause effects in various vascular structures of the body. It's extremely powerful to actually dump actual adrenaline directly into the bloodstream. So this is one of the most important, probably, synaptic connections on the sympathetic side. So again, this preganglionic neuron is cholinergic. It happens to have synapses inside of the medulla that synapse with chromaffin cells. The chromaffin cells themselves, of course, have cholinergic receptors because they have to be able to receive information about acetylcholine, but they themselves actually produce adrenaline and noradrenaline, or epinephrine and norepinephrine. Let's go ahead and look at our other exception on the sympathetic side of things. We have a preganglionic neuron that is blue, cholinergic. We have a postganglionic neuron that is blue, cholinergic. And in this particular specialized example, the specialized exception, both of the neurons produce acetylcholine, and in this case, it causes the sweat glands to be activated. And so we'll just draw a simple little, I don't think we'll even label everything. We'll just go ahead and draw a presynaptic neuron that is releasing acetylcholine, and the postsynaptic neuron, which in this case is actually cholinergic as well, it has receptors for that acetylcholine. So cholinergic neuron, cholinergic receptors, and the same thing at the very end of this. These sweat glands have cells that are glandular epithelial cells, regular old epithelial cells, but they happen to have the ability to secrete sweat. So you don't want to be secreting sweat all the time. So you only want to do that under conditions that they are induced to do so by these neurons from the sympathetic nervous system. And therefore, these postganglionic cholinergic cells will input acetylcholine at certain times. And of course, these epithelial cells, these specialized epithelial cells, will have cholinergic receptors. So they will be able to receive the cholinergic input and then therefore be activated when that postganglionic neuron is activated. Finally, let's look at the way that most of the sympathetic nervous system works. We have a preganglionic neuron that is blue. We have a postganglionic neuron that is red. And both of these, by the way, both of the upper examples that are shown here have short preganglionic neurons and long postganglionic neurons. We discussed that in the previous video. All right, so in this case, we have a cholinergic preganglionic neuron and an adrenergic postganglionic neuron. So let's go ahead and add that in there. The preganglionic neuron releases acetylcholine at the synapse. The adrenergic neuron releases norepinephrine at the synapse. As we said, norepinephrine is quite similar to epinephrine. It has a similar molecular structure and it has similar effects on target organs. So let's go ahead and show this first synapse here. We have a preganglionic neuron that is blue and releases acetylcholine, no surprise. The postganglionic neuron, however, we're drawing in red, and therefore that neuron actually is adrenergic, and its own product is going to be norepinephrine, but it actually has cholinergic receptors, so we have to actually draw those, label those in blue. But at the other end, so at its receiving end, it receives acetylcholine and is activated that way, but at its sending end, at its axon terminal end, it actually releases norepinephrine primarily, and therefore its targets are going to have adrenergic receptors. And we're going to go ahead and pull one of these out and give a specific example. So here we have a preganglionic neuron that is blue, cholinergic, and we have the postganglionic neuron that is red, adrenergic, short preganglionic, long postganglionic. And take a look at this. This particular cell happens to be synapsing with the exact same cell, the exact same group of cells that we had drawn earlier from the other side. Remember that a lot of actual tissues have dual innervation. We're just showing that. We're showing a specific example. So it turns out this same autorhythmic cell in the SA node, which sets the whole pace for the way that the heart is going to be beating, beats per minute, and to a certain extent the contractility of the heart or the strength of each contraction, is going to be enhanced from the sympathetic side because there is an adrenergic receptor which receives norepinephrine from adrenergic neurons. So this is a specific and I think very beautiful example of dual innervation where we're looking at really at the cellular level exactly how that would work. Yes, it's possible for a cell to have both types of receptors. The receptors are just proteins that are created within that cell's nucleus. You happen to have genes that are activated that allow for cholinergic receptors to be created, transcribed and translated, and then inserted in the membrane. And then we also have the ability for a gene for adrenergic receptors to be transcribed, translated, and inserted in the membrane. And so depending on what is needed at the moment, we have a great deal of control coming from the ANS in a decision about exactly how our heart is actually going to be behaving. If we're at rest, the parasympathetic side is in charge and is going to be slowing down that heart rate. But if we're excited or if we're exercising, our heart rate is going to speed up because the direct synaptic input comes from the sympathetic side. Hey, let's take a look at those adrenergic receptors. So we said there's this specific adrenergic receptor on the autorhythmic cells of the heart, and then most every other sympathetic target is also going to have adrenergic receptors, with the exception only of the sweat glands and then those specialized cells within the adrenal medulla. So let's talk about the adrenergic receptors: alpha 1, alpha 2, beta 1, beta 2, beta 3. So very briefly, each of them has their own purpose and distribution around the body. So we're going to give a few specific examples. This is not an exhaustive list, but here we go. Alpha 1 receptors are found on the blood vessels, especially on the walls of the arteries, where they allow for vasoconstriction of the blood vessels in certain circuits. We'll talk more about blood vessel diameter and vasoconstriction. It's quite important down the line. Alpha 2 receptors are also found on the blood vessels, and they actually tend to inhibit the same thing we were just talking about. So alpha 1 receptors tend to cause vasomotor tone, which is where they kind of tighten up the blood vessels, kind of cause a little bit of a squeeze on them. Whereas alpha 2 receptors will actually cause the vasomotor tone to be relaxed a little bit. If a person has high blood pressure, one of the things they might be given as a prescription might be some kind of an alpha 2 agonist, which the term agonist means it enhances the activity of something. So it enhances the way that the alpha 2 receptors are receiving information. And so alpha 2 receptors will tend to cause the blood vessels to relax a bit, and that in turn causes the blood pressure to go down. All right, let's go ahead and talk about the very famous beta-1 receptors. Beta-1 receptors are the ones that we drew up above on the walls of the autorhythmic cell there on the red side of things. The beta-1 receptors, they enhance your heart rate, they enhance contractility of the heart, or the squeeze of the heart as it is contracting. And therefore, a beta blocker, which is a very famous pharmacological class of drugs, beta blockers will, to a certain extent, quell this activity. Most cardiac patients will actually be on beta blockers because oftentimes, as part of the cardiac disease, they may have some issues where the heart rate is a little bit higher than optimal. And therefore, a beta blocker will cause, the beta-1 receptor causes enhancement of the heart rate, the enhancement of the contractility. So a beta blocker will inhibit both of those things. Slows down the heart, stops it from squeezing quite so hard. Now, the beta-2 receptors will be found on cells of the lungs. They allow for the dilation of the bronchioles. So these are important in times when a person maybe is suffering from anaphylaxis or anaphylactic shock, and their airways are actually closing up. The term dilate means to open things up. So constrict is closing things together, but dilating is opening them up. So generally speaking, during anaphylaxis, one of the most worrying symptoms of anaphylaxis is that the bronchioles start to kind of close down, and it's harder for a person to draw breath into their lungs. So it turns out that adrenaline specifically, one of the famous things, or epinephrine specifically, like the EpiPen, one of the big things that it does is it causes those bronchioles to open up by interacting with those beta-2 adrenergic receptors. And the beta-3 receptors are found in two interesting spots, in the gallbladder, which it attaches to the liver, and it actually stores bile that is produced by the liver. And then the urinary bladder, which of course stores urine that is produced by the kidneys. We have some idea that in the urinary bladder, the beta-3 adrenergic receptors help to relax the walls of the bladder. But we actually don't know exactly what's the purpose of these receptors in the gallbladder. So there we have it. We have 5 different types of adrenergic receptors, which have very different roles in different parts of the body depending on where the receptors are found. And I think it can be kind of a confusing lesson because it's hard to conceptualize how one particular neurotransmitter can have so many different effects on different structures in the body. And the same with the cholinergic side of things. How can you have the same substance, acetylcholine, having these very different effects, either excitatory or inhibitory, depending on the structure that they're actually interacting with? And that's kind of a confusing story, but I think that's some good work to be done in this class. Not just thinking about how a neurotransmitter is released, but thinking of how it is received. That's the part of the story that I find that students have the hardest time embracing. So pay attention to those receptors. In terms of pharmacology, in terms of the way that drugs are prescribed and that they interact in your body, the receptors kind of tell the whole story of which drug is appropriate at a certain time. So it's not just the release of a substance that's important, it's how a particular organ or a particular tissue is receiving it and reacting to it.